Alright, welcome back everyone. Let's go ahead and move on to our second video focusing on photosynthesis. Before we actually start talking about photosynthesis though, let's talk a little bit about the nature of light and why that's going to be important for better understanding what's going on later on when we actually do talk about photosynthesis. So, light that comes from the sun delivers to us all different types of light or electromagnetic radiation. And the different types of light we receive differ in what we call their wavelength. Now, if you take a physics class, one of the first things that you'll learn about light is that light behaves both as a particle and as a wave. In our regard, we are talking about the wave-like properties of light. So, when you're talking about a wave of light appears to us in consecutive waves, and light travels, of course, very, very, very fast, with these waves coming towards us very, very, very quickly. So how we define light is based on its wavelength, the distance between two consecutive points on consecutive waves. So usually we use the crest of each wave or the trough as each wave as our basis for that. So the closer the waves are together, the shorter the wavelength, the further they are apart, the longer the wavelength. If you actually look at the spectrum of different types of electromagnetic radiation, very short wavelength light would be things like gamma rays and X-rays, and very long wavelength light would be things like microwaves and radio waves. So the light that we can actually see with our eyes actually falls within a very narrow spectrum, which we call the visible light spectrum. It runs wavelengths from about 350 nanometers to about 750 nanometers. You'll hopefully recall that nano, the prefix there, the SI prefix for the metric system, means 10 to the minus ninth. So we're really talking about light that has very, very, very short wavelengths in the grand scheme of things, but we'll talk more about that here in just a little bit. So what I want you to understand here is that there is a relationship between a light's wavelength and the amount of energy that light has. As the wavelength gets shorter and shorter, the light starts to contain more and more and more energy. So gamma rays and X-rays have a lot more energy than any of the types of radiation you see towards the right. So the downside there is that if you're talking about extremely short wavelength radiation, since it has a lot of energy, that type of radiation can potentially harm living cells, such as what you see with UV radiation and the sunburns people get every summer. That is evidence that the cells of your skin are being actively harmed by the high energy radiation you receive from the sun. On the other side, long wavelength radiation has a lot less energy. So if you take radio waves, for example, there are thousands of radio stations throughout the country that are constantly broadcasting radio signals, and those radio waves are constantly bombarding our tissues on a daily basis, but it doesn't do us any sort of harm because that type of long wavelength radiation has practically no energy associated with it, so it's not really possible for doing us any harm. If you focus in on the visible light spectrum, again, somewhere between 350 and 750, you can actually see that we can split that up into different colors. The longer wavelengths are responsible for kind of the more purple and indigo colors, and then as you go up towards blue and green and yellow and orange, and then finally up to red, that shows you the wavelengths getting longer and longer. Now, visible light, if it's a mixture of all of those wavelengths, will appear white to our eyes. So that's why we sometimes call it white light, if it's a mixture of all the wavelengths in the visible spectrum. But if you're able to isolate just one particular wavelength, that light that you see will have a particular color to it. If you put a filter over white light, like a red filter or a blue filter, what those filters are actually doing is preventing all the other wavelengths from getting through, except those corresponding to the color that you actually want to see. So the reason why we bring this up is that photosynthesis is going to make use of enzymes that catalyze chemical reactions, and photosynthesis is also going to use proteins that are involved in capturing the energy of light by complexing with pigments that only absorb very specific wavelengths of light. So understanding that light comes in all different varying wavelengths is actually a very important thing for us to get out of the way. So the idea here is that if you're talking about photosynthetic pigments, whether it's either chlorophyll A or chlorophyll B, or the accessory pigments, which are called carotenoids, Any wavelengths that are not absorbed by these particular pigments, and you can tell which wavelengths are absorbed based on where your peaks are in the absorption spectrum, any wavelengths that are not absorbed, the kind of more flattened out portions, are going to be reflected instead. And this is actually very interesting because this will give us insight into why different objects appear certain colors to us. If we take photosynthetic organisms, for example, plants, algae, and cyanobacteria, the reason they appear green to us is because if you actually look at the absorption spectrum for chlorophyll, both chlorophyll A and chlorophyll B, but together, chlorophyll A and chlorophyll B do a good job of absorbing your purple and violet and indigo colors, so you can see we get good coverage over on this side of the spectrum. And they also absorb kind of the more orange and red colors, but look at what color they do not absorb, the green colors. So this explains why chlorophyll appears green to our eyes. The chlorophyll that is found in photosynthetic organisms absorbs the colors that we do not see, the purples, the blues, the reds, the yellows, and the oranges, but they reflect the colors that we do see, which is the green colors, and those wavelengths of light are specifically reflected back at our eyes, hence the reason why these organisms appear green to us. Now, if we actually take a look at the molecular structure of these different pigments, chlorophyll A, chlorophyll B, and then carotenoids, if you're curious as to why pigments absorb different wavelengths of light and why some chemicals do not absorb visible light, I'm probably not the best person to explain that to you. That's a lot of very complicated organic and physical chemistry. But the basic idea is that it's the chemical bonds within a pigment that allows that pigment to absorb quanta of light, specifically different wavelengths. Usually it's going to be carbon-carbon double bonds, aromatic rings, and carbonyl functional groups that tend to absorb light the best in the UV to visible range. Now, again, I'm not going to pretend to be any sort of expert on that, but if you actually want to look at the differences in the absorption spectra for chlorophyll and chlorophyll B, chlorophyll A and chlorophyll B, which this picture here is not the best thing to look at, so let's back up just a minute and look at this one right here. Chlorophyll A, its absorption spectrum is plotted out in a dark green trace, and then chlorophyll B is in a much more light green trace. You can tell that the absorption spectra are very different. They behave very differently in how these two pigments absorb light. But compare and contrast the molecular structures of chlorophyll A and chlorophyll B. Literally the only molecular difference is that chlorophyll B contains an aldehyde functional group right here, whereas that functional group is not present in chlorophyll A. That one seemingly very minor substitution in the grand scheme of a molecule that looks almost identical is enough to shift the absorption spectrum of A very differently into chlorophyll B. So that little difference is enough to produce very different behaviors in how these two pigments absorb light. And then, of course, the carotenoid pigment that you see right here looks very different, but it does contain a lot of carbon-carbon double bonds, which, again, that's one of the ones that is good at absorbing light. Now, here's an interesting question for you. Why do you think leaves are green during the spring and summer, but then reddish-orange during the autumn? If you actually look at the absorption spectrum for these different pigments, again, we'll look at the one that's a little easier on our eyes. The chlorophyll pigments absorb pretty much everything except the green colors. So it stands to reason that during the spring and summer months, when leaves appear green to us, the chlorophyll pigment is the major pigment found in the leaves, because that's the major thing that we're seeing. But compare that to the carotenoids. The carotenoids absorb light fairly well in the purple range, very well in the blue and the green range, but look at what the carotenoids do not absorb. They do not absorb the yellows, the oranges, or the reds. And those just happen to be some of the prettiest colors for us to look at as the leaves turn in the fall. So a possible explanation for this, if we didn't know anything else about how plants work as they transition from season to season, it would almost seem that the chlorophyll disappears as the summer months end and the carotenoid pigments hang around. And that's actually not far off from the truth. Okay, finally, let's continue and wrap up this discussion by discussing the color of objects. So the idea here is that if you take visible light and separate it out by wavelength, you will actually see a rainbow of colors. After a rainstorm, this is actually the very reason why you might see a rainbow. The light from the sun, which is usually all kept together and appears as white light, passes through rain clouds, and the rain droplets actually act as a spectrum and separate the light out into all the different wavelengths. So, as we've said, the fact that objects appear certain colors to our eyes is actually evidence that those objects contain pigments that absorb certain colors, which again, those are the colors we don't see, and then reflect others back at our eyes, which are the colors that we do see. So, on two kind of extreme ends of the spectrum, if an object actually absorbs all the wavelengths and reflects none of them, then the object is going to appear black to us, since black essentially represents the absence of light. So if you're looking at a black object like a shirt or a cabinet or a TV, that black paint actually is made of a type of pigment that absorbs all of the wavelengths of visible light. On the other hand, if an object appears white to you, it's the exact opposite. White pigments actually do not absorb light. They reflect all the colors back at us, and that is why they appear white to us, because white, as we said before, is a combination of all the colors at once. Now, something you can kind of do on your own, but I can kind of push you in the right direction here if you want. What would the light absorption spectra look like for pigments that you find in the following objects? So some examples: the skin of a red apple, so it obviously appears red to us, a person's blue jacket, a pumpkin, which obviously appears orange, and then kernels of corn, which appear yellow. Now, I'll leave you to your own devices on kind of figuring this out for yourselves, but again, if you go backwards and you actually look at what an absorption spectrum looks like, first, start by thinking about what that color of that object actually is. Is it red? Is it blue? Is it yellow? Is it orange? And then match up the color that it actually looks like, and then try to create your own absorption spectrum so that you get big peaks in the colors that your object is not, and then a big valley in the area in which your object actually does appear that color. So that'll be a good little exercise for, I think, you to do on your own. Okay, so finally, let's talk a little bit about the process of light absorption. So when an organic pigment, like the chlorophyll and carotenoids that we've seen before, or the retinal, which is a vitamin A derivative that absorbs light in our own retinas, as we've seen, they can only absorb a very specific range of wavelength and energies. So what happens when these pigments are exposed to wavelengths that are not within their specific range? So if these pigments absorb a wavelength that is greater than the specified range, then that light is going to lack a sufficient amount of energy necessary to excite electrons in the outermost shell of the atoms of that particular pigment. In other words, if the wavelength is too long, it's not going to that light doesn't have enough energy to do what it's supposed to do. If these pigments absorb a wavelength that is lower than the specified range, then that light obviously has enough energy, but the problem is that there is too much energy, so that extra energy is actually going to damage or destroy the pigment through a process that is called photo-bleaching. This is actually something that you may have experienced before if you accidentally look at the sun for too long or for any time at all. You actually start to feel a little bit of pain, and you kind of close your eyes and you kind of see nothing but bright light. That is evidence that the pigments in your retina have been damaged by the sun, and it's going to take a little while for your cells in your retina to repair the damage. So you won't really kind of stop seeing that bright light until your cells can bring in new pigment and replace the old damaged pigment. Okay, and then something that is actually a little bit more poignant for studying photosynthesis in the lab is I want to introduce a device which is called a spectrophotometer. A spectrophotometer is a device that allows scientists to measure the amount of light that is absorbed by a particular compound or molecule. So if you know that you're dealing with a photoactive molecule or pigment, you know you're dealing with something that absorbs light in the visible spectrum, or you know that you're dealing with something that has a color to it, you can actually use a spectrophotometer to measure how much light is actually absorbed. So the idea is that the spectrophotometer contains all different types of filters that can take in white light and then split it up into only one particular kind of wavelength, and then pass that particular wavelength of light through a sample. Usually it comes, the sample is contained within a little plastic cuvette, which contains a solution in which your molecule is dissolved in. So the light passes through the sample, and then there is a detector on the other side of the light that takes in the light and measures what percentage of the light that came in through the cuvette actually makes it through to the other side. So if the molecule in the sample does absorb light, then only a fraction or a percentage of that transmitted light will be detected, and that amount of light absorption is actually something that can actually be quantified. I'm not going to get into the actual nitty-gritty of this process or how it works. We might actually get to discuss that in the future when we start talking about biotechnology. Okay, that's going to do it for this video. Thanks for joining us, and next time we will actually start talking about the light-dependent reactions of photosynthesis, and then in our last video we'll wrap up by talking about the Calvin cycle and the dark reactions. Thanks again, see you next time.